Should people on the live stream be seeing my blinking screen? Yep. Okay, good morning, everybody, and welcome to today's Saturday morning lecture, which is the first of 5 live stream events that we will provide this spring. Today, our speaker is, my name is Dr. Stanley Yen. I will moderate today's session, and because of COVID, our public lecture series has moved online. So today, our speaker, Dr. David Sivak, who is an associate professor of physics at Simon Fraser University. He's the holder of a Canada Research Chair Tier 2 and the winner of the 2020 Young Investigator Award from the Biophysical Society of Canada. In the live stream description, you will also find links to learn more about our speaker's career and accomplishments. Your questions for the speaker may be submitted via the chat box on the live stream. So now I invite David to speak to us about the ingenious nanomachines inside all living things. David?
Okay, thank you very much. Thank you for the organizers for inviting me for this and putting together this great recurring series. And thank you for joining us this morning. So my name is David Sivak, and I'll be telling you today about some of the ingenious nanoscale machines that nature has evolved inside all living things, some of the interesting functions they accomplish, some of the challenges they face, and nevertheless, some of the remarkably impressive performance they can achieve. And by the end of this talk, I hope you have some idea about why these molecular machines in their operation face challenges that amount to bicycling in a cyclone and swimming through maple syrup. So I'll start with basically a physicist's view of life at a very high level. And one of the central conundrums from a physicist's perspective that living things face. And I'll summarize this by observing that the second law of thermodynamics is incredibly inconvenient for living things. What do I mean by that? So the second law of thermodynamics, one of the central principles in all of physics, says that entropy or disorder increases over time. This is the reason why when you put your earphones into your pocket, back when we had wired earphones, and you take them out a few hours later, they have a tendency to be tangled up. This is why even your most careful Lego creations tend to evolve into a mess of individual Lego pieces on the floor that you step on. When you look at biological systems, you notice that in fact, there's an incredible amount of order. Living things are really predicated on not having disordered systems, right? You look at a cell, the individual building block of essentially all living things, and it's an incredibly ordered system. There are very, very specifically specified compartments that have very particular functions. Dynamically, it behaves in a very orderly fashion. It's really central to living things that they are ordered, right? That there's something that is you, that has a stable shape over time, that does not dissolve. And so it's really fundamental to living things that they're fighting all the time against this second law that says that disorder increases. So from a physical perspective, this is one of the central challenges that living things face. How do you maintain order in the face of the constant propensity of the world to become more disordered? And at some level, there's no great mystery about why this is. It's because living things have evolved to siphon energy off of energy flows that are present in the world around them. And so at the very highest level, the way that living things accomplish this is by siphoning energy flows off of energy flowing from the sun. So the sun shines high energy light onto the earth, which is eventually then re-radiated away. But in the meantime, living things have learned to absorb energy from the sun and make use of that energy to do productive tasks, to build themselves up, to create order. At a much smaller level, individual organisms, such as this schematic of a person here, take in food and output waste products, and in the process, siphon off energy from this food as you process it to do useful things. And at the most fundamental level, the way that this energy is siphoned, the way that energy is converted from one form to another and stored is through molecular machines. So molecular machines are protein complexes. They're a few nanometers in size, typically. A nanometer is a billionth of a meter. So these are very, 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 very small objects. They're composed of proteins. This is the typical biomaterial that most molecular machines are made of. And they convert between various different forms of energy. So they convert between mechanical energy, often stored in the compression or the stretching of various materials in the same way that you can compress or stretch a spring. They convert between mechanical energy, electrical energy, separating charges across distances, like in your electrical outlet, chemical energy stored in high energy chemicals, and then perhaps more subtly in the concentration of different molecules or different materials in one place or another, rather than a uniform distribution of materials. So these are all the different kinds of energy modalities that molecular machines convert between to be able to accomplish things for living things. So what I'd like to do now is I'd like to take a bit of a tour through some of the ingenious machines that natural selection has managed to evolve, and really some of the really remarkable functions that nature has figured out how to accomplish with these molecular machines. So at the most initial level of energy harvesting, energy siphoning from the sun are the machines that actually directly receive energy in the form of sunlight. So these are present in all photosynthetic organisms, such as plants, various kinds of bacteria that can accomplish photosynthesis as well. One of the primary examples is what's known as photosystem 2. It's a molecule that's embedded in the membrane, shown here in gray. Typical size around 10 nanometers in scale, again, just as a reminder of how tiny, tiny, tiny these systems are. And the way this works is the sun shines, light is absorbed by this machine, and it converts that energy in the form of that light that it absorbs into pushing charges apart, separating positive from negative charges across the membrane that it's embedded in, essentially creating a voltage difference, or you know, you can even think of it like a battery. So what this system is accomplishing is it's converting sunlight energy into electrical energy, which is a different form of energy that then other machines, other systems within living things can make use of. So really what you have here is you have an analog of a solar panel. Nature has figured out millions and millions of years before humans figured out how to make materials that would absorb and harness energy from the sun. Another example of the class of machines that have been evolved is what are known as rotary motors. These are machines that have whose function involves rotation of some crankshaft within their makeup. My favorite example is one of the central machines in all of energy processing and all of living things. It's known as ATP synthase. It's a molecule that synthesizes ATP molecules. Again, typical scale, about 10 nanometers. Again, embedded in membranes. The way it functions, it makes use of a voltage difference that has been established by other machinery across the membrane it sits in. And it takes advantage of that electrical energy to drive rotation of a central crankshaft. Rotation of this crankshaft in turn pushes on this red subunit below and drives it to make ATP molecules out of ADP molecules. And ATP molecules are very high energy, but they're very stable, they're very long-lived, and they're very portable, they're very small. So basically what you've done here is this machine converts electrical energy into portable chemical energy. So you can really think of this at some very meaningful level, like essentially a factory making batteries that then can be taken elsewhere and then used to power other processes. Another ingenious rotary motor that nature has evolved that's used in many, many different kinds of bacteria is what's known as a flagellum. It's basically a long hair that's then embedded, anchored in the outer membrane of bacteria. Again, its typical width is something like 10 nanometers. The hair itself is actually much longer than 10 nanometers, but its scale in the membrane is around 10 nanometers. Again, a nanometer, a billionth of a meter. And the way this machine works is, again, it makes use of an electrical, a charge difference across this membrane to drive rotation of a crankshaft within this membrane. And that rotation then rotates the entire hair. And if you zoom out to look at the entire bacteria with this hair extending away from it, rotation of this hair induces a propulsive force in the fluid that the bacteria is in, that it lives in. These bacteria live in water, and pushes in a particular direction. And this is actually how these bacteria navigate. This is how they move from one place to another to avoid prey, to seek out food, etc. So what this flagellum, this rotary motor, accomplishes is it transduces, it transforms electrical energy into motility, into self-propelled motion of the organism itself. So you can really think about this as being a reasonable analogy with the propeller on the outboard motor of a powerboat, right? It's rotating, pushing the water away from it, and thereby driving the organism in a particular direction. Another fascinating class of these machines is what I'll call transport motors. One example is shown here in this artist's depiction of what we think these motors are roughly doing. It's known as kinesin. It has two little feet that walk along filaments within cells. You can think of these as roads, transport roads, taking you from one place to another within a cell. I'll try playing this again. And it latches onto cargoes on the other end. And so what it accomplishes is it transports these cargoes to locations they're needed within cells. It goes in particular directions along these roads. You can think of these as directional roads, and thereby concentrates cargoes where they're needed for other purposes. Another example of such a transport motor is known as myosin. Again, both of these roughly at the scale of 10 nanometers in size. It travels along different filaments, different roads within cells. These are known as actin filaments. The names are not so important, accomplishing essentially another modality of transport. So what these transport motors are accomplishing basically is they are converting chemical energy. They actually burn ATP, they consume ATP molecules, these high energy molecules, and they're turning that into motility, into self-propelled motion, in their case, transporting cargoes that they're attached to desired locations. So a really interesting analogy for these motors is you can really think of them like car engines, burning chemical fuel to transport you to a particular place. Some other interesting machines that nature has evolved. Here is an example from viruses. This is a particular virus known as Phi29. The gray object here is a shell, a hollow shell that the virus produces when it's still inside its host. And then it must package its genetic material, in this case DNA, inside this shell so that it can then exit the host and then find some new host to infect. Again, roughly the size is something like 40 nanometers in size of this entire virus, so still very, very small. And there is a very strong motor that is embedded right at one end of this shell, at a pore in the shell, and its job is to spool the DNA inside this viral shell, tightly compact it, because the shell is very small compared to the material that it has to accommodate, So it has to push against very high pressures, load all this DNA in, and then seal off this pore so that the virus can then leave the host, retaining its genetic material. And in fact, the pressure is so large that it actually creates a spring-loaded mechanism. When the virus infects a new host, it then forcefully ejects its genetic material into the host, and that's actually how it can overcome some of the host defenses. This machine also consumes ATP in order to push the DNA against the pressure that it faces. So you can really think of this machine as transforming chemical energy into essentially a high pressure container with important valuable contents. So at some level, a rough analogy is that this is like a factory that's making these joke canisters where you open a can of nuts and out pops some snake, some fake snake in your face. It's a loose analogy. Another fascinating class of machines is what are known as pumps. These are proteins which are embedded in membranes again, and their job is to redistribute small ions, small molecules of sodium and potassium across the membrane. So they consume ATP, they consume chemical energy in order to push sodium ions in one direction, potassium ions in another direction, that are then used by other processes within the cell. So again, what they're doing, they're consuming chemical energy, they're transforming chemical energy into concentrated, into concentrations of various molecules in particular functional places. One more large class of these machines that I find really fascinating as well, that are really central to the core aspects of living systems, which is the copying and the translation of their genetic material into the basic biomaterials that compose living things. One core example of this is the machinery that copies DNA. It's known as DNA polymerase because it polymerizes, it creates a new DNA strand complementary to an existing DNA strand. Again, the machine itself, shown here in green, roughly 10 nanometers in size. And its job is to copy the DNA so that organisms can reproduce and divide. Another example is RNA. RNA is a substance that is made by reading from the recipe encoded in the DNA genetic material. There is machinery that creates RNA, again, by reading along a DNA strand, using that recipe, that code, to make an appropriate RNA molecule. So that machinery, again, roughly 10 nanometers in size, is shown here in blue and green, wrapped around an orange DNA double helix. One other central actor in these kind of processes is known as the ribosome. It's essentially the translator that translates RNA, which is itself a code of some sort, a recipe, into proteins. And proteins are the central actors in most biological processes. They're kind of the real action. You can think of DNA and RNA as, blueprints, recipes, and then protein is often where the actual stuff happens in biology. So the ribosome's job is to actually make these proteins from RNA. They're slightly larger, something like 20 nanometers in size, but again, roughly the same scale. Very, 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 very small. In addition to all these individual machines, nature has also evolved many ways in which machines work together in cooperative, collective assemblies. So one example is this rotary motor, this ATP synthase that makes high energy ATP molecules. You can really think of this as two machines coupled together, one of which, shown in blue, takes this voltage, this electrical energy, and transforms it into rotational motion of a central crankshaft. The second piece, shown in red, that transforms this rotational motion into high energy, chemical energy in the form of these ATP molecules. Many of the machines that are used to copy and translate that we just talked about on the previous slide are actually composed of several different machine components, each of which have specialized tasks within the overall goal of copying and translating this genetic material. So for example, the ribosome is composed of several different components, some of which are involved in translating in proofreading, in bringing the relevant materials to the location, etc. In terms of our transport motors, they rarely act in isolation. Typically speaking, any given cargo that needs to be transported will have multiple motors attached to it, some of which are working together. They're of the same kind of motor, trying to transport the cargo in one direction, such as these set of green motors on the bottom here, and often antagonistic motors that are trying to transport the cargo in opposite directions are attached as well. And it's through the interplay between these different motors, regulating how many of these motors are attached at any given time and how active they are, that living things can regulate where their cargoes are taken within the cells, which directions to transport them along these cellular filaments, along these little roads for navigating the cell. And finally, perhaps the most well-studied, best understood, in some ways very ornate collection of machines is in muscles. So the basic function of muscles is to contract and thereby to rearrange the relative orientation of different limbs in your body. And so the way that muscles are constructed is in red here down on the bottom are a collection of myosin motors. These are cousins of the motors that transport cargoes along cellular filaments, but these are now stationary, fixed in place on their own short filaments, but they're opposed to these purple filaments that are actin filaments, exactly the roads that these motors usually transport cargoes along. But these juxtaposition of these different filaments allows the motors to take individual steps. And when many of these motors all take individual steps, the effect is to compress the muscle, to shorten the muscle, and thereby accomplish the function of that muscle. So it's only through really the tight coordination of all of these different machines and the tight arrangement of them in this very regular array that the machines work in a functional way. So this has been sort of a tour of the various different functionalities of the kinds of machines that nature has evolved. There are many other examples. I've limited myself to some of what I find to be the most ingenious and really some of the most central to all living things. What I want to talk about now is I want to talk about, in some sense, from a physical perspective, the strange world that these machines inhabit, the unique, distinctive set of challenges that they face. In the previous section, I've tried throughout to make analogies between these microscopic nanoscale machines and human engineered machines that seem to accomplish very similar kinds of functionality. What I want to talk about now is distinctive aspects of these machines due to their size and the materials that they're actually made of. So I've tried to remind you throughout the previous section of the rough size of these machines. Let's take a little bit more of an explicit, careful thinking about exactly how small these are. So again, I tried to draw analogies between these molecular scale machines and human scaled machines, for example, transport motors and a car engine. A car engine is typically speaking roughly a meter in size, maybe a little bit smaller. If I go down a factor of 100 in size, I get to a screw. That's typically about a centimeter in length or width. I go down a factor of another 100 in size, and I get to the typical width of a human hair, roughly 1/10,000th of a meter, roughly 100 micrometers or microns. I go down another factor of 100 in size, and I get to the typical size of bacteria, typically 1 micrometer in width for your average bacteria. I go down even another factor of 100, and I get to molecular machines. So 10 nanometers, 0.00000001 meters in size. These are 100 million times smaller than car engines, even though at some very abstract level, they accomplish similar goals. So given that, they experience a very different kind of world than a car engine does. And I'll try to unpack that a little bit in the coming slides. One more aspect I want to focus on. Here's a reminder. visible light, the wavelengths of visible light, red, yellow, green, blue, and so on, are typically a few hundred nanometers wide. That is much larger than these molecular machines. These machines are so small that light is too big to resolve the structures of these machines. That gives you some sense of what we're working with here. These are incredibly tiny machines. So let's talk about one other aspect of them that's very distinctive from human engineered machines, and that is the materials that they're built with. So let's start with one of the hardest substances known to humans, diamond. I'm now measuring things, the elasticity of these materials, essentially how compressive or how stretchable these materials are. The technical term is the Young's modulus, but the terminology doesn't really matter. Diamonds are incredibly hard. It turns out they're roughly 1 terapascal in their elasticity. But what's important is the relative stiffnesses of different materials. So if I go a factor of 10 floppier than diamond, I get to steel. Steel is a typical material used to make human scale machines. I go a factor of 10 floppier still, and I get to typical stiffnesses of wood. Another factor of 10 even floppier, and I get to the typical stiffness of plastic, like the plastic in your milk jug. And even another factor of 10 floppier, and I get to the typical stiffness of rubber. Proteins, again, the materials that molecular machines are composed of, typically lie somewhere in the range of plastic to rubber in terms of how stiff, how compressible, how stretchable these machines are. So molecular machines are composed of materials that are 100 to 1,000 times floppier than the materials that we typically make machines of. These are by no means rigid objects. So let's think about what the ramifications of this are. So one way to kind of back up and think about what results from these properties of machines is to think about what's known as the phenomenon of Brownian motion. When you look in the microscope at objects that are a micrometer in size, and this was first done roughly 200 years ago, looking at pollen grains in water, what you find is you find something like this. This is a microscopic image of roughly micron-sized, micrometer-sized, millionth of a meter pollen grains. These are just sitting in a microscope slide in water. You're not directly moving the system around. These pollen grains continuously jiggle around. They never stop bouncing around. And at the time, this was an incredible mystery for people. What is the source of the constant jiggling of these microscopic objects? They tried all kinds of different materials to see what would happen. Everything jiggled. They even took little shavings from the Sphinx in Egypt to look at, and everything jiggled when they put it under the microscope in water. And it took roughly 100 years for people to figure out really what was at root the underlying source of this. And among other people, Albert Einstein was one of the people who really came up with the definitive answer and demonstration of what was the source of this constant motion. So let's zoom in from this micrometer scale to a much smaller scale. Let's go another factor of 100 or 1,000 smaller and see what's actually going on. And so here, this is now a simulation. The previous slide was an actual microscope video. This is a simulation of what is going on with physically realistic dynamics. The red represents individual water molecules, roughly 1/4 of a nanometer in size. And this blue sphere is meant to represent some small protein, maybe a nanometer in width. And so what you observe when you look at the dynamics of these, again, this is way too small to directly observe with light. You need other methods to actually see these kinds of things, is that this blue sphere is constantly being pummeled by kicks from the water molecules. It's constantly bouncing around due to these constant yet irregular collisions with the water molecules. So if I have an object that's roughly a micron size, the reason why it's continually bouncing around is because it's being continually kicked by water molecules that you can't directly see. So that's really the ultimate source of this constant jiggling. Any objects are constantly being jostled, pummeled, and kicked by the surrounding molecules. The water molecules are far too small to see. And when you get to a size of a micron and smaller, that jostling is actually quite significant. So objects that are of that size or smaller, and recall that molecular machines are a factor of 100 smaller than a micrometer. They're typically 10 nanometers. They're even more violently jostled by these collisions. So a molecular machine is constantly randomly bouncing around, constantly randomly jiggling. It's taking steps in all kinds of directions. The steps it wants to go, the functional directions, but also side steps, back steps, and so on. So in some very meaningful sense, molecular machines are trying to bike in hurricane strength winds, in cyclones, like this image from The Wizard of Oz. They're trying to make progress in particular directions, along particular roads, in the face of constant violent jostling. So this is a fundamental challenge they face. So when people try to make more faithful depictions of what these motors are actually doing, they start making videos like this. This is meant to be kinesin, the same system I showed earlier in a video, traveling along a filament, traveling along a road. And this is meant to capture more of this random aspect to its behavior. Sure, on average, it's going in one direction, but that is layered on top of an incredible random component to its motion. So any motion accomplished by a molecular machine is only in a particular direction on average. It's going to be taking steps in all directions. It's going to be rotating in all directions due to this constant random jostling by the molecules that surrounds it. Moreover, if we think about machines that are composed of assemblies of individual machines, the connections between these machines by necessity must be incredibly floppy. You cannot have rigid connections between nanoscale size protein machines. It's simply not physically possible. Another central aspect of these machines functioning is that they have no inertia. Because of their incredibly small size, because of the crowded environments they actually find themselves in, surrounded by other molecules that again are too small to be directly observed with light, they're constantly, and because of these constant violent kicks from their surrounding molecules, they're always being frictionally damped. Any step in a particular direction is immediately damped out. There's no persistent motion of a molecular machine. So in this very real sense, machines move through an incredibly viscous surroundings. It's the analogy of a human swimming through a pool of maple syrup. And because of that, every motion of these molecular machines is reliant on these random kicks to actually push it along. It cannot coast through any particular stage of its machine cycle. It cannot rely on persistence of velocity to get it anywhere. So again, it's absolutely obligatorily reliant on these random kicks to actually take each of its steps to achieve its outcomes. Finally, there are no temperature differences accomplished by molecular machines. And this is really in a striking contrast to human scale machines. If you think about human scale, for example, power plants, the way they actually transduce energy, this could be geothermal, this could be fossil fuels such as coal or other fossil fuels, this could be nuclear energy. is you heat steam, and then you have a temperature difference between hot steam and cold water. And the spontaneous energy flow from the hot to the cold system is used to generate energy, to turn a turbine, to generate electricity, etc. This is not available to molecular machines. They're so small that they cannot maintain any temperature differences. They simply cannot generate energy fast enough to maintain any sort of persistent temperature difference. So they really have to operate in fundamentally different ways from the kind of machines that humans have created. So given these set of challenges, you might think there's no way these machines can actually accomplish anything useful. And nevertheless, they can. And what I want to tell you about now is I want to tell you about some of the remarkable functionality that these machines actually can accomplish, that nature has evolved them to actually do. One question you can ask is, how efficient in energy conversion are these machines? There are many different kinds of machines, as I've tried to convince you, and their efficiencies vary widely. But what I'll focus on in this section here is some of the most impressive examples to give you a sense of what is possible using these molecular machines. And again, my favorite machine, ATP synthase, that creates these high energy chemical bonds, can reach 90% efficiency in its energy conversion. And you compare this with a typical car engine that has about 20% efficiency in conversion of gasoline to motion of the car. Another measure you can ask about is, how rapidly can these machines operate? What's the speed with which they can actually function? So this flagellum can rotate as fast as 300 rotations per second. That turns out to be faster than the turbine in a jet engine. An incredibly fast operation. The machinery that copies DNA, the DNA polymerase, there are a variety of different polymerases in different organisms for different functions, but the fastest polymerases can copy DNA at 1,000 base pairs per second. Look at these transport motors. The fastest known transport motors are a particular kind of myosin that can transport its cargoes at 7 micrometers per second. That doesn't sound very large, 7 millionths of a meter per second, but when you consider the size of these machines, that works out to roughly 100 body lengths per second. That's the equivalent of me running 400 miles per hour. These machines can be incredibly accurate, thinking about the copying and the translating machines. So the ribosome that translates RNA into protein, it's making a sequence of amino acids based on the recipe from the RNA. It makes one error, it incorporates one incorrect amino acid in every 10,000 that it strings together. The machinery that makes RNA from DNA makes even less errors. It makes something like one error in 100,000 RNA bases that it transcribes from DNA. With RNA and protein, if you make an error, you can just throw away the faulty RNA or the DNA or the protein and start over. What's a much more fraught operation is when you're copying DNA. If you make an error in copying DNA, and that faulty copy then goes into your offspring, you've now lost the information about what the original sequence was. This is what's known as a mutation. It's important that there aren't too many mutations. So the DNA copying machinery is unbelievably precise. It makes roughly one error in 1 billion base pairs that it copies. Okay, so I've told you some about the impressive capabilities of these molecular machines. I want to conclude today by giving you just a brief introduction to my own research group. Everything I've talked to you about so far is relatively well established. I can tell you a little bit about what we do as a research group, how we fit into all of this. And I'll tell you one very brief story about things that we have studied in relation to these machines and their capabilities. So here's the group, a set of very capable graduate students and postdoctoral fellows. We're in a physics department, but we come from a variety of backgrounds and we use a variety of different tools in our work. We're a very interdisciplinary group, making use of physics, biology, mathematics, computer science, engineering insights, as well as chemistry in our work. And it's really this interdisciplinary aspect, I think, that is, really important to making important advances in these kinds of areas at the intersection between different fields. The kind of questions that we're interested in asking are things like, what are the physical limits on what these molecular machines can accomplish? How efficient can they be? How accurate? How speedy? And so on. What kinds of designs achieve these physical limits? What kinds of engineering principles govern the optimal operation of these molecular machines? And when we go out and look into what natural selection has evolved to accomplish these kinds of tasks, do these evolved machines, do they actually reach or do they near these physical limits, such that we can understand in some sense what evolution has been driving them towards? And finally, can we take these ideas, can we take these engineering principles that we devise in the context of studying machines, and can we help engineers who are making new nanotechnology for applications like efficient computing, renewable energy harvesting, as well as, for example, targeted drug delivery? So I'll tell you one short vignette about a recent study of ours to give you a flavor of the kind of research that we actually do. So we thought a bit about this idea of the very floppy connections between machine components. Again, because these machines are so small, because they're connected by they're composed of relatively soft material, you can't rigidly glom two machines together. They have to have relatively soft connections between them. And what we found through our theoretical work is that floppy connections between machines can actually be advantageous. Intuitively, you think, if I have a floppy machine, how in the world can it actually work effectively? But it turns out there actually can be advantages to this. And the basic intuition of this is that, again, because these machines rely on being randomly kicked each step of the way, having floppy connections allows individual components of a machine to be kicked separately. So if you think about the different pieces of my rotary ATP synthase as these blue and these green dots, they're sitting in some landscape, sitting at the bottom of a hill and waiting for some random kick to jump you over the next hill to your next stable valley. If the two systems if they're rigidly linked, you need to kick them both over the hill simultaneously. You need to have a relatively large random kick. If they're flexibly linked, then you can kick one subsystem, one component of the machine to its next state, and then you can sequentially kick the other subsystem over. And it turns out that two small kicks occur much more frequently than one large kick, just due to the nature of how the water molecules are colliding with these systems. And so the upshot is that because it's so much easier to get two small kicks than one large kick, by having flexible connections, it actually can result in faster operation, in faster speeds for these machines, which is a relatively counterintuitive result that we're now thinking further about the ramifications of this. Finally, let me acknowledge funding sources for our research, the kind of public support that makes what we have learned about these machines that actually makes that possible. My group is primarily funded by NSERC, the federal Canadian organization funding natural sciences and engineering research, as well as the Canada Research Chairs Organization, another federal group. And we do a lot of high performance computing to simulate and to analyze the theories that we devise. And so Compute Canada is the federal Canadian infrastructure for this. And also thank you to you as public taxpayers. You may not know it, but a little bit of your tax dollars are supporting this kind of research. And so thank you for that support. And finally, I'll wrap up by giving you a few ideas of places to go if you'd like to learn more about some of these ideas, if you find these intriguing thoughts to pursue. Here's a set of YouTube videos. You can just type into YouTube to find larger, more extended videos of the kind that I showed you a couple snippets of today. to see artist depictions of how these machines actually operate. Here's a set of books that are cast at a very general audience, accessible, non-technical level, that nevertheless get into some of the core ideas that we're really thinking about in the context of how these machines operate and what they're capable of. And finally, here is a relatively accessible textbook that really focuses on just the simple scales of biology. So things like how fast can living things operate? How big are the different components? How strong are they, and so on. So a lot more details of the kind that I told you about today. So with that, I'll wrap up. I thank you very much for joining me today, and I look forward to hearing some of your questions.
Okay, so thank you very much, David. I invite members of the audience to submit questions to the speaker on the YouTube chat channel. So maybe I can start with one question. Which one of the nano-machines that you have described does the COVID virus most similarly behave like?
It's an interesting question. I am by no means an expert on COVID. So COVID itself is an organism itself. It presumably makes use of some machines in its operation, but it also, in some sense, hijacks a lot of the machinery of the host organism that it infects. And so, what's quite likely is that during much of its life cycle, it's actually making use of a lot of these core biological machines that are used, for example, in copying of genetic material and transcription of genetic material. But I can't speak in great detail to particular machines that are specific to COVID, and that it's not just kind of piggybacking on within, for example, the human cells that it infects.
Okay. Another question is, you said that these machines evolved by natural selection. Anywhere do we see primitive versions of these nanomachines which could be steps in the evolutionary history of these nanomachines?
So essentially kind of missing links or common ancestors. What I would say is that these machines are so widespread that the evidence is very, very strong that there were, common ancestors that then diversified within, among different organisms and even within different organisms to accomplish different tasks. One thing you can see is that in some sense, we can see how nature has evolved different versions of similar machines. And one of the tricks that it uses is it uses identical building blocks and then puts them together, for example, in different numbers. So this ATP synthase, this rotary machine that makes ATP, it has a certain rotational symmetry in that it's made of essentially different trivial pursuit pi pieces that are then brought together to create cylindrical structures. And in different organisms, there are different numbers of those different pi pieces. And that leads actually to interesting effects like a different gearing ratio, a different leverage between the voltage and the chemical energy that it produces, which actually appear to have evolved to essentially match the strength of the voltage in different organisms. So I don't know that I can point to a specific example of a primitive machine, but you can certainly see how nature has used essentially modular architecture of these machines to create different versions to accomplish different goals. Another example is that researchers are now monkeying with these machines and trying to learn how to recalibrate them, how to reorient them to create different functionalities. And so a lot of progress has been made in the last few years on identifying what are sort of the architectural details that produce one behavior rather than another. One example is these myosin motors I've talked about. A given myosin motor goes in a particular direction along these cellular filaments within cells, along these actin filaments. But you can make minute changes, minute mutations to a given myosin, and you can actually flip the directionality. And so in some way, we are now uncovering the ways in which evolution can make small changes to these machines and alter their behavior in functional, productive ways. And if you're interested in these kinds of questions, I'd point to this third book here by Nick Lane, which is looking at this question of energy transmission in sort of primordial life, and thinking about how can you have very primitive systems that can nevertheless do functional energy harvesting. In his case, he's focusing on geothermal sources, underwater geothermal sources, and thinking about how can very primitive versions that are much simpler than the versions that we see now, how could they have been sufficiently effective to allow reproduction of life and then further evolution to create the more sophisticated, robust versions that we see now.
Okay. Now another question is, could engineers copy anything from the principle that you learned from nano-machines to engineer things on the macro scale? Are there new engineering principles that we could apply?
On the macro scale?
Yeah.
So that is
machines for everyday life.
Yeah, that is an interesting question. So people are very actively looking at how do we take insights from what nature has evolved and make use of them to figure out functional nanotechnology. On the macro scale, again, because of some of these differences that I really focused on in I guess the second section of the talk, there really are vast physical differences in the behavior of these different systems. So for example, because of the random operation of these transport motors, it's the equivalent of if you got into your car and every third time you put your foot on the gas, it went backwards, you'd make a pretty different car. But fortunately, cars are made out of stainless steel. They're made out of just the random component is negligible in their operation. That may not feel like that when your car breaks down, but believe me, compared to these molecular machines, it's negligible. So a lot of the insights that we worry about are things that simply don't apply on the macro scale. So I personally do not know of specific examples where people have taken these insights. In some ways, we've already had 150 plus years to think about how to engineer macro scale systems. So we're way ahead on that front, and we're really making only the barest advances, only the kind of first baby steps to really think about how do you engineer on the micro scale. So I think we're not yet at the point where we can take these insights. And there's also this question of just how do you transfer when it's very different physical regimes that these things are operating in.
Okay, one last question. What about mitochondria? Given what we know, how different are these versus the other nanomachines that you showed us?
So mitochondria, you can think of it as almost like a housing for molecular machines themselves. So the thing that makes mitochondria the energy powerhouse or the power plant of the cell is that they are studded with millions of these ATP synthase molecules. That's what makes the mitochondria the energy producing hub. There's a variety of other machinery in mitochondria that then kind of creates the necessary conditions for these machines to do what they do. So in some sense, what I've been telling you about is I've been telling you about a story of energy transmission at the tens of nanometer scale. You can zoom out a bit and you can ask about, well, at the level of, say, a micron, the typical size of a mitochondrion, what is the story of energy transmission? And it is, in some sense, the collective behavior of all of these machines that are packed within mitochondria that then gives the mitochondrial function itself. So they're very closely related. They're not distinct questions, and one in some sense emerges from the behavior of the other.
Okay, and a question just came in. What specific profession would explore the details of molecular machines and change them into something that humans can use on a non-molecular level?
What profession is involved in exploring these things and trying to figure out how to move forward on creating new ones?
Yeah.
I guess it goes under a variety of guises. I mean, a lot of the work is being done in academic settings right now. And so these are people who might be bioengineers, nanotechnologists. To the extent that these ideas are beginning to get traction, they're beginning to move into industry as well. So people who are thinking about drug delivery mechanisms, perhaps in pharmaceutical companies, people who are thinking about renewable energy in national laboratories or in particular technology companies. That's probably where this kind of work is being done. A lot of this work is at early stages, so I suspect most of the people are going to be primarily on the academic side at the moment. But as these ideas begin to demonstrate their utility, I think these are increasingly moving into industry.
So if a high school student were interested in this and wanted to pursue a career in this, what kind of things should they study? Biology, physics, what?
I think I think the key is to pursue your passions. If this really interests you, these ideas are at the intersection of physics and molecular biology and chemistry to a certain extent. Engineering insights are very useful in what we do. From a practical perspective, we spend a lot of time, doing math, doing computer programming to work out the consequences of the ideas that we propose. So really a broad-based scientific exposure, I think, is really the way to go. And there are many different angles from which you can bring insights to really learn about this and uncover new ideas.
Okay, I think that's it for the questions. So again, I would like to thank David for this very interesting and enlightening lecture. And our next lecture will be on February 6th, and you can find the link for that on the YouTube channel, Triumph Lab. So hope to see you all again next month. And